action potential arrives, calcium channels open, right? Calcium flows into the neuron.
Yeah.
Right? So that leads to the release of acetylcholine from the vesicles. And acetylcholine binds to the receptors in the sarcolemma.
Yeah.
Those receptors on the sarcolemma are channels, correct?
Yeah.
These channels allow sodium to flow into the cell and potassium to flow out.
At the same time?
Yep.
Or, okay.
Yep. So we're going to call it cation channels. So
Okay.
either sodium or potassium can get through, but sodium concentration in the extracellular fluid is higher.
And that's why it's deep.
So sodium diffuses into the cell.
Okay.
Potassium concentration inside of the cell is higher. That's why it diffuses out of the cell.
Okay.
Good? However, flux of potassium is lower than sodium. So that leads to depolarization. You good?
Yeah.
So that depolarization is called end plate potential.
Okay. That last part with the channels opening and stuff, that's end
This
plate.
generates end plate potential. End plate potential is restricted to the neuromuscular junction.
Okay.
If end plate potential crosses the threshold, the action potential on sarcolemma is generated.
Okay. But only if it crosses.
Only if it crosses threshold.
Okay. If
Only.
it doesn't.
If it doesn't, there is no contraction.
And it's just relaxed.
Just nothing happens actually.
Okay.
Okay. Now, how is the action potential formed? If
This is if contraction happens.
Don't run again in front of the train.
Okay.
Let's just talk about action potential, okay? So if end plate potential crosses the threshold, sodium channels that are voltage gated,
Okay.
okay, open. Okay?
On the sarcolemma?
In
That's
the
the
sarcolemma.
Okay.
When they open, membrane potential becomes more positive, depolarization,
Okay.
right? When it reaches the peak, voltage-gated sodium channels close and voltage-gated potassium channels open. So potassium starts to go out, and that's repolarization. Sodium-potassium pump restores the concentrations.
Okay.
And you have the propagation of the action potential all the way through the sarcolemma. So that's going to be action potential on sarcolemma. Action potential on the sarcolemma, which is T tubules. Okay. Now DHPR and RYR, two receptors. Here, they are connected. Just makes some kind of pretentious picture. You don't need to know the names of the receptors. I don't really focus on that. what you have to understand, it's going to be T2. Okay.
What's the other one? That one.
This?
No, I know that's T2 bolt. That one, yeah.
And sarcoplasmic reticulum.
Okay.
So what happens when action potential reaches those receptors? Receptors open calcium channels. I try to color code them. Calcium channels on a sarcoplasmic reticulum, and calcium flows out into the sarcoplasm. So far.
What is that part at the top called? Is that just...
Which one?
Like, does that have a certain name? Like, is that the coupling thing or no?
This one?
Yeah.
This graph.
Yeah, and the pumps and stuff. Does that have a certain name?
That is excitation.
Okay.
This is all excitation.
Okay.
Muscle gets excited. And then calcium release couples it to the contraction.
Okay.
Does that make sense?
Yeah.
Good?
Yeah.
No, it's good. We're recording something. Is that understood?
Yeah.
Now, here you have myosin.
Only if calcium's released, though, right?
Calcium is released.
Yeah.
Getting to the next step. And actin. Actin is blocked from interaction by tropomyosin. However, on tropomyosin, there is troponin. Calcium binds to the troponin. That's calcium. It binds to the troponin. Sort of separates tropomyosin from actin, which now can form cross bridges with myosin.
Okay.
And the contraction ensues.
Okay. So the thing with tropomyosin and myosin and all that, that's the coupling, though?
That's the contraction. The idea of excitation, contraction, coupling. So the word coupling means that one process requires another.
Okay.
Which means contraction cannot happen without excitation. And excitation results in the contraction.
Okay. And then the cross bridge happens once that all happens?
Once this happened, you have cross bridge cycle.
Okay. Can you go over cross bridge like that? Like, draw that out?
I can try. So you've got actin, you've got myosin, and you've got cross bridge. Cross bridge at this point has ADP and phosphate bound, and it is cocked back, it is charged, it has energy. Does that make sense?
Yeah.
When ADP and phosphate are released, the cross bridge propels the actin molecule forward. And I don't know how much forward, it's noticeable, but I hope it is. Okay? So you can see kind of, sort of. that the actin molecule moves forward. Does that make sense? Then, the next step, you need ATP to release actin from myosin cross bridge. So at this point, myosin cross bridge is sort of broken, dissociated, because myosin head binds ATP. Next, on color. Well, we ended up with 3 steps. Yeah, it should be fine. I mean, they have a separate step for, I'll try to repeat it. So then you have the breakdown of ATP into the ADP and phosphate. Does that make sense? Which is still bound to myosin. And the last step, it forms cross bridge again.
And it doesn't stop unless you run out of calcium
and it doesn't stop as long as you have ATP and calcium.
Yeah.
Just do this, separate them. Okay.
Okay.
Better?
Yes.
Awesome.